Hello, Amin and I are presenting the Taurus One study today, which examines the safety of magnetic brain stimulation for people living with multiple sclerosis. So first, what is myelin and what does myelin actually do? So the yellow cell here is a neuron that is coated with myelin, which is in green. So myelin is an insulating layer that wraps around the neurons and protects it. Myelin is very important because it allows the electrical signals to travel along the neurons quickly and reliably. However, in multiple sclerosis, the immune cells, which is in pink color, enters the brain and attack myelin. This results in myelin loss. This also affects the electrical signals don't reach where they need to go. And ultimately, people end up with coordination problems or cognitive problems or other problems associated with multiple sclerosis. So, if we can find a treatment to add myelin back to the brain, we can probably reverse the damage from MS. So that's when we come to the idea of myelin repair therapy. So people living with multiple sclerosis have also identified that myelin repair is one of the biggest unmet needs because currently there is no treatment to promote remyelination. So that is why our team is currently trying to develop a treatment for myelin repair. And one promising approach that we are using is magnetic brain stimulation. So what is magnetic brain stimulation, MBS for short? So MBS involves holding a copper coil over the head, and then within the coil, there is an electric current running through it, which then generates magnetic field. So this magnetic field can further induce another electric current running in the brain tissue underneath the coil. And this induced electric current can change the electrical signals where the cells used to communicate. So this is the first time that our team is using MBS as a remyelination therapy. So how might it work? So the white cell here is the cell that can make myelin. So when myelin damage occurs, myelin is lost and that cell is also lost as well. Yep. And then the immature cells can respond to it. They can migrate towards the injury site. However, they can't make myelin. So this is where the MBS comes in. The MBS increases the cell activity. So the immature cells can mature into the myelinating cells. So they can add more myelin to the brain. So these laboratory findings that help us to move into the clinical stage involving people with MS. So in 2021, we completed the Taurus One study with a small number of research participants. And the primary objective was to determine whether MBS is safe and tolerable for people living with MS. We also obtained the preliminary data about the effects of MBS on the MS symptom severity, such as fatigue, depression, anxiety, quality of life, and also obtained the preliminary data on the myelin repair based on MRI scanning as well. So the Taurus One Trial was conducted at the clinical research facility at the Medical Science Precinct in Hobart in Tasmania. So 20 people living with MS were recruited in the Taurus One Trial, and they were randomized into either MBS group or placebo group. So in placebo group, the participants underwent the same procedure, but no real stimulation was applied. Before the treatment, participants completed the MRI scanning and also the questionnaires on the MS symptoms. After that, the participants attended 20 daily sessions from Monday to Friday over 5 weeks, and each session lasts around 15 minutes. Then, right after the treatment, participants again completed one MRI scan and the surveys on the MS symptoms again. And 3 months post the treatment, post the last treatment, participants were asked again to complete the surveys. And now Amy will continue to talk about the results of the Taurus One.
Hello everyone and good afternoon. I hope the lunch was good. So I'm going to talk about the results, including the adverse events, which is our first and primary aim of the study. So as you can see in this table, the baseline characteristic of the participants, 20 participants who participated in this study, in terms of the sex and duration of MS diagnosis, which in terms of the statistical calculation is no different between these two groups. And in terms of the level of disability is measured by a standard measure of disability in people living with MS, which is EDSS. They were slightly different between the MBS group and placebo group. And this was sourced from the mild disability, the EDSS between 2 to 3.5, which in MBS group has more people assigned to this level. But as this is not the main exposure of this study, this slight difference is not going to make any difference in our study outcome. In terms of the MS time, they're fairly similar apart from the primary progressive MS that we had 4 participants in the MBS group. And this is the main and primary result of our study, which is the safety outcome. As you know, an adverse event is any untoward medical occurrence that may happen in any research study in participants who receive the treatment, which can be any intervention like medication or what we have here, magnetic brain stimulation. But these adverse events are not necessarily related to the treatment, and it could include even accident, getting COVID or flu-like symptoms, or the adverse events that we were looking for, such as headache, seizure, that as part of the defined adverse events related to MBS. But fortunately, none of the study-related adverse events, which we call adverse reaction, none of the participants reported in this study. But we have several adverse events, including fractured arm post-fall, shingles, worsening of memory and balance, and generalized rash, which based on the neurologist in the study, neurologist decision, none of them were related to the intervention. As Tram said, we also had some secondary aims in this study, including a series of questionnaires, surveys, and also we used a mobile application called Floodlight Open to assess the cognitive function in participants in this study. I brought here one of the results from the Floodlight test, which shows the number of steps before, after 20 sessions and after 3 months. Although the differences were not significant, but as you can see, in blue color shows the MPS group, which fairly kept the level of baseline or before treatment number of steps during the study, but the placebo group slightly deteriorated the number of steps. These are preliminary results. We are not saying that our treatment 100% effective. This is an important thing to be noticed here. But this is one of the things that we are going to look further in the next stage. In terms of depression and anxiety symptoms, we use the scale to measure the severity of symptoms reported by the participants. And another important point here is that all these are the average symptoms or average severity in each group. In none of the groups, the average is above the standard or clinically significant depression or anxiety. So these are within the normal range that even myself, which I'm not clinically diagnosed with depression, might report these level of symptoms. And these two groups were not significant in terms of comparison of the level of anxiety and depression. Another scale was fatigue severity scale, which again, both groups reported the same level of fatigue symptoms during the study and 3 months follow-up with no difference, and the same as quality of life. Now we are going to the most important secondary aim of the study, which are the MRI outcomes. So as you know, MRI is an important indicator of disease activity in MS and play a role in diagnosis of MS. And MRI scans shed light to what happened in the brain during the MS pathology. In our study, between the two groups of MDS and placebo, none of the participants had any new or enlarging lesions between the beginning and at the end of the trial. In terms of brain atrophy analysis, as you know, brain atrophy is a natural process of aging, but in certain medical conditions, the most famous one is dementia, and also in MS, we can see accelerated and early onset of brain atrophy. Again, all the participants in our study had less than 2% change. This is the change before and after the treatment in brain volume between baseline and follow-up MRI. And there was no difference between two groups. But as Tram clearly described, the process of multiple sclerosis affect the white matter or myelin content of the brain. We were specifically interested in seeing what changes that this treatment, magnetic resonance stimulation, would affect the myelin content of brain. QT value is inversely correlated with the myelin content in the white matter. And if you remember the picture that Tram shows, the device placed in the frontal side of the brain, which is called frontal lobe. So the myelin content in the frontal lobe is specifically very important for us to see whether is there any change or not. So as I said, this is a preliminary result. We didn't see any significant difference between two groups, but in placebo group, in gray color, the placebo group continuously lost the myelin content. So, but in the intervention, in the treatment group or MPS group, there was no change, and it remained stable. The myelin content in time 1, which is the pre-treatment, and time 2 after the end of the treatment. And in other words, the placebo group continued to lose myelin over the course of the study, while the MPS group maintained myelin at the baseline level. And these are the list of lovely group of project contributors who worked from the conception of the study and during the study. And especially on behalf of our team, I would like to appreciate all the participants who kindly contributed their time to this study, and our project funders, the list are here. Thank you for the event contributors today and the funders of this event, and thank you so much for your attention. So, continuing the presentation with this question, you may ask yourself why we need to go to Taurus 2, why we need to stage the clinical trial. So, this is a good figure to show that for any clinical trial or any treatment or any medical procedure to be proved efficient and safe, there is a valid and standard process, what we call phases of clinical trial. Tram explained a bit about what happened in the laboratory and what Kaylin and her group in the Galileo research team conducted heaps of research on cell line and animal models, and saw that this MDS treatment is safe and effective in generating myelin and keeping myelin alive and supported by the nerve cells. So this is a typical study from lab to the clinic. So with the help of the MS Flagship clinic side and the lab side, we designed the phase 1 of the study, which I've already reported the result. We found it is safe, but this is not sufficient. We need to prove that this is efficient as well. This would change, or this would affect positively the indicators or outcomes that affect the people living with MS. So we need to recruit more participants to be able to prove the efficiency of this method. So the phase 2 study requires more participants, which in a standard pattern, it requires hundreds of people. Now I would like to invite Professor Bruce Taylor, MS Neurologist and the clinical lead of MS Research Flagship and the principal investigator of the Taurus 2 study.
Thanks, Amin. Tram and Amin have really summarised, people. Sometimes one of the considerations is that you make a discovery in the lab, it will immediately work on humans. Why do we have to go through all this sort of rigmarole to prove that it works? Because you're not talking about something that's take a tablet, has no side effects. You're talking about something that people have got to come in every day for 4 weeks and have a stimulation. It's also expensive. The equipment, you need people, and you need to know that it works, and you need to know it's safe. And MS has been played for generations by people doing an experiment on 1 or 2 people and saying it works, and then when it's done on a larger scale, it doesn't work, and it can be harmful. And we don't know, and we don't know whether this works. Our preliminary data, which Amin and Tram have presented from the lab and from phase 1, would tell us that there's a good, that we've got enough information to move this trial to the phase 2, which is a much larger trial, which is going to be rolled out hopefully very soon, and includes Launceston, and Lauren Giles is here who will tell you about Launceston's involvement in this trial. It's also going to be run in a number of sites in mainland Australia. So, what we're going to do is basically do the same thing we did in Taurus One. People are going to be enrolled who have MS, who've got a particular disability level, and I'll describe that a little bit in a few moments. It's going to be running these 5 centres and possibly another centre on the mainland. We are hoping to recruit 120 people with MS, and we're going to enrol them to have either real stimulation or sham stimulation. And one of the important points from the first trial was that we showed that Amin and Tram can give people sham stimulation, and the people who did not know whether they were getting the sham or the real, they couldn't pick it. And so that's really important. So the only person who knows whether it's the sham or real is Amin and Tram, but they're not involved in the analysis of the data. So it's a blinded trial. So it's really important, and that's a critical point, because being in a clinical trial, people want it to work, and they have a large placebo effect. So we could do anything to people in a clinical trial, and we could find a meaningful difference from them from baseline to the final time point. But that doesn't really mean anything, because that could be placebo. And what we need to know is that what we're doing is safe, tolerable, and not only that, but effective. And that's the purpose of a phase 2 trial. So basically, we're going to do exactly what we did in the Taurus trial. We are aiming to recruit people in 5 sites. We're able to, it says 108 up there, but we were hoping to really get to 120, because some people in all sorts of a clinical trial like this will have a degree of attrition. We're going to compare functions beforehand, afterwards using MRI, essentially the same thing, but we had to change one of the markers that we used in the first trial, which is called the floodlight, because it's no longer available. The company that makes it, one of the drug companies, has taken it out from research for reasons we don't understand. So we've replaced it with a marker called the Multiple Sclerosis Functional Composite Score, which basically does the same thing, but doesn't do it as neatly as it does it very neatly, but doesn't use a mobile phone app to do it. We're going to particularly look at the MRI scans, because the MRI scan is actually showing that we're adding, if it does continue to show what we showed in the first part, show that we're adding myelin. So again, now people will have 20 sessions. We'd like them to complete that in 4 weeks, Monday to Friday, daily for about 15 minutes, and then have 2 MRI scans. The main outcome measures will be again, it's going to be safety and tolerability. Were people able to get in, have the study, have the treatment, and then we're going to be very interested to know whether it affected the MSFC, which is a multiple sclerosis functional composite score, which is made up of a timed 25-foot walk, a 9-hole peak test, which some of you may have done, where you've got to actually pick up 9 peaks from a dish and put them into a grid and take them out of the grid and put them back in the dish. Do that in both hands. It's a measure of manual dexterity. Then we're going to do a test of cognition, which is called the single-digit modality test, which is again a very well validated way of looking at processing speed within the brain. Now we're hoping that these will show the same trends that we saw with the first phase of the study. We're also going to do the same MRI measures that we did in the first part of the study, and they will be analysed centrally at the Sydney Neuro Imaging Assessment Centre. So what are the inclusion criteria? Now, it's always ageist clinical trials, I'm really sorry about that, but 18 to 65, we're older than most people, most trials, but above 65 people get other reasons for cognitive decline. They don't necessarily respond as well because of the natural ageing of the brain, and so we have to try and select a group where we'll see the most. If it's going to work, we'll see the improvement. They must have MS, obviously, which is also very important. You know, they must be stable and not have had a relapse recently, and also must be stable on treatment. And they must have an expanded disability scale of 1.5 to 6, which means they've got some disability, so we can actually measure change, but they're not so significantly disabled that we're not going to be able to reliably measure change. So 6 means you need to use a cane for most activities of daily living. So, and then, again, they must be willing to participate, which is incredibly important. You know, it's hard work, and one of the biggest advantages we had was that Amin and Tram was so fantastic with everybody, and people would start a conversation, and they'd have this conversation for 15 minutes a day for 4 weeks, and then we're disappointed that they didn't have to come in and finish the conversation, which is fantastic. And you know, people must have capacity to provide consent and be willing to and able to participate in all follow-up assessments. Exclusion criteria. One of the important things is you've got to be able to have an MRI scan, so you can't have any metal in your body which will prevent you having an MRI scan. You know, the people who are pregnant or wanting to become pregnant, we can't give an experimental treatment, even though I sincerely doubt it will have any effect, but that's just one of the things that you can't do in a clinical trial. Now, there's certain things people, if you have stimulation of the brain, you know, there is a slight risk of causing a seizure. And again, the type of transmission, the stimulation we're giving, makes it very, very unlikely. But people who've got a history of seizures, or stroke, or brain surgery, or bipolar or mania, because they can actually be upset by magnetic stimulation. Claustrophobia is an important one because you can't have an MRI scan if you've got claustrophobia. Significant head trauma. Again, there's and uncontrolled migraine, and it is also one. But some of these things, substance abuse is also another issue. But in some of these, there is discretion that if people get infrequent migraines, or they're very well controlled, or they've had a past history of substance abuse, then we can allow them to be in the study. And if they have had minor head trauma, it's not an issue, but we just need to be aware of that as the investigators. Also, some medications which may affect the way MBS affects the brain, particularly tricyclic antidepressants in therapeutic dosages that we would use to treat depression, not in dosages which people will use to treat bladder dysfunction. And there's a number of other medications, antipsychotic medications, which do affect the way the brain responds to brain stimulation. So if you enroll, we'll ask you about what medications you are on. Again, you've got to fit into the EDSS range and the age range. And if people have previously had brain stimulation, which is sometimes given for things such as depression, we can't have people in study because they'll know that they're not having the real thing. So, and they must be English literate or have someone who can provide appropriate translation and or if they're currently involved in another interventional study or investigational study, we can't have people in those two. So how to get involved? Well, in Launceston, Dr. Lauren Giles is going to give us a few words. She's the neurologist in charge in Launceston. What?
There is a section you jump.
Sorry?
There's a section you jump.
Oh,
Yeah, this is what I'm sorry.
Oh, the
But
project.
previous one.
Yeah, sorry. So these are the people who contributed to the project, and particularly I'd like to really emphasise that what we're doing is based on the work that Kayleen Young's group did in the lab. This is, groundbreaking, innovative, world-leading research has come from the laboratory, and now we're taking it to the clinic. And this is the sort of research we want to be doing in Tasmania. This is research which is, as I said, groundbreaking, internationally significant research. And as you see, there's quite a number of people there, and I'd like to particularly mention Carly Cohen and Mark Hinder, who's our TMS specialist from works at the School of Psychology, Kayleen, Natasha Stevens, who's been instrumental in getting this work off the ground, Angela McKechnie, who designed the database, Julie Sampson, who is the new coordinator for the Taurus 2, Vincent, who's our clinical research fellow, who's heavily involved, Amin and Tram. Particularly like to thank the funders of this project, which is the Australian Government Medical Research Future Fund, particularly the Irene Phelps Charitable Trust, and also the Clifford Craig Foundation. And in Launceston, I'll let Lauren thank the people in Launceston.
So just one section remains. During this study, we had the opportunity to talk and spend time with a number of lovely participants who devoted their time, and after a couple of weeks became our ambassadors and inviting their friends and relatives to the study. So one of those lovely people is Meg Denham, who's a trial participant and person living with MS, and also one of the members of the Consumer and Community Friends Committee. And we had discussion before, and I asked her to share her experience in the study, and she kindly accepted. So, Meg, can you please describe a bit your experience? No, this
one working?
Yeah.
Okay. So my experience, I was diagnosed with MS in about 2010. When I was first diagnosed, you probably wouldn't have known that I had MS, but gradually my mobility has dropped off. I can walk less well. I've tried lots of different disease-modifying therapies over time, but still I'm going downhill. So then I came to this trial.
So how did you hear about the study, and why did you decide to participate?
So I'm part of the Consumer and Community Reference Group, and so that was put to us as being a really interesting trial. I thought that sounds fantastic. As well as that, there was a great presentation last time we had one of these, and that absolutely convinced me. It seemed so interesting and such a good thing to be a part of.
And how was your experience during the study, the 20 sessions and 3 months afterward?
Well, I would say I was one of those people who just enjoyed being with you, part of it. It was lovely to come in every day. It was a very small impost. Quickly come in, get my head zapped over, ear zapped over there. There was an MRI, there was some little tests. It was a very easy process. As I say, the support from getting in, getting it done, very easy every day. And of course the people were lovely, so that helped.
And my final question is, we had a discussion before, and you said it is important to participate in clinical trial for the MS community. So what's your opinion? Can you please share this with us?
Yeah, I think it's not just important, it's really vital. It's the way that we get these really great ideas from research actually out to the people who benefit from us, and that's us. And so if we don't do this, they just stay in the lab. People think that they're a great idea, but what we really need is we need these processes, these treatments to come out to the people who will benefit from them, and that's us. And so the way we get that to happen is that we participate in the trials. We give a little bit of our time, and out of that we all benefit fantastically. And I think this is a really worthwhile one particularly.
Thank you. Thank you so much.
Oh, thank you, Amin.
Hello everyone, my name is Lauren Giles. I know many of you, but for those of you I don't know, nice to meet you all. I'm going to take up just 1 minute of your time, just inviting local Launceston participants to be involved in this trial. So we're very grateful to Bruce and the team for asking us to participate in this trial. I give lots of talks to medical students and junior doctors about MS, and I always finish with a slide about remyelination and about how this has long been seen as the holy grail in MS land, and it really is. So if these sorts of this treatment or other medication that are also being tested for remyelination work, then we're really getting close to a cure for MS. So I think this is incredibly exciting. It's incredibly exciting for us here in Launceston as well. It's our first toe in the water since I've been here in clinical research. We hope it paves the way for lots more north-south collaboration. I'm also grateful to the support of the Clifford Craig Foundation. We're lucky here in Launceston that we've got a sort of a fundraising body within the hospital called the Clifford Craig Foundation, who support a lot of hospital-based research, not just through funding but through the ability to access extremely skilled clinical trials nurses, some of whom are over here today. So Monica and Shelley will be leading the Taurus trial from a research nurse perspective, and my sidekick and partner in crime, Karen Lewis, will also be involved in recruitment of people. So just flying the flag for Launceston. This obviously is a trial where being nearby the trial centre is key, because you're coming in every day for a month. So if you're interested, we're not actively recruiting at the moment, but we're very happy to take names, and we'll get back to you when we're ready to go. So please participate. Thanks.
So I'm a geneticist, and I study the genes involved in MS. Now, inside all of us is a brain and a spinal cord, and what you can see in this picture here is that brain and spinal cord are the areas that are damaged in the development of MS. But when we zoom in at a closer level, down to the cells inside the brain and the spinal cord, there's a lot that's going on. We have the immune system crossing the blood-brain barrier into the brain and attacking the nerve cells to degrade that myelin sheath around the neuron that we've heard a little bit about before. With these several different cell types involved in the development of MS, what I'm interested in is something that's common to all of them. And that is called DNA, or collectively, the genome. So what is a human genome? Well, it's basically a code of letters, A, C, T, and G. And inside each of the cells inside your body, there are 3.2 billion letters spread across something called chromosomes they're packaged in. So 3% of this genome, about 3 million coding letters, are genes. So they have some kind of functional role in your cells and your body. If that entire genome was to be printed out, it would fill 120 books, 1,000 pages at 4.5 font. It would take you years and years to read the human genome. And some people in the UK did this, and it's part of the Wellcome collection. And you can see, you can't because the font is so tiny, but that is what the human genome looks like. So there are 2 genomes inside each of us. One from mum, one from dad. And that's how genetics works. They combine together to make a new person. As a geneticist, we represent this slightly differently, and this is called a pedigree. The squares are males, the circles are females, and together the 2 genomes are halved and recombined to make their offspring or their child. And then the next generation, if that child goes on to have children, half and half again to make the next generation. So why is genomics important? Well, genes control our basic biology, how the body works, how we respond to drugs, our height, our eyesight. Everything about us is controlled by DNA. changes to the genome make you who you are. So between all of us, we share a lot in terms of our genome. It's all very similar, but there's about 0.1% difference from each other. So Des and I, at around 3 million places in our very long genome book, we are different to each other, and that's what makes me taller than Des. But changes in your genome can also cause disease and influence how you respond to different drugs or treatments. So we study those changes to understand why diseases develop or occur and how to improve health. To study those changes, we do something called genome sequencing. So genome sequencing is a technology that allows us to read that genome book. We have a reference genome. It's sort of like the most commonly assembled book that represents the most common human, and that is 3.2 billion bases. We compare each person's genome that we sequence to that reference book and find the differences. So those DNA changes, and around 3 million sites is where they'll be different to that reference genome. We then study those differences, focus in on them to look at how these differences may be causing disease. So to sequence a genome, we take a person's book, we extract DNA from cells, from blood, and what we essentially do is shred that book up into tiny little pieces. And then we build it back together against the reference genome at the top, realign those pieces of the book to recreate the book of you. So for multiple sclerosis, we know that the risk factors can be put into two main groups. One's an environmental group and one's a genetic group. So the environmental component of MS includes things like Epstein-Barr virus infection as a risk factor, vitamin D, sun exposure, and things like your weight or smoking. The genetic component of MS is something we can study using genome sequencing. And for a large part of the genetic study, genetic component, we know the factors that contribute to MS. But half to 3/4 of the genetic risk of MS is unknown. And that is what I'm interested in. MS is a complex disease, and finding out those unknown genetic factors is going to tell us more about how MS develops. So to find those unknown genetic factors, I study families. So around 1 in 4 to 1 in 6 people with MS has a family history of MS. Families with multiple people with MS are fairly uncommon, but they do occur, where 2, 3, 4 people in the family all have MS. From a scientific perspective, members of the same family share the same environmental risk factors that probably have all been exposed to Epstein-Barr virus or similar sun exposures. They're from the same part of the world, so they have the same ancestry background, and they also share the common genetic risk factors for MS. That lets us study rare genetic changes that may only be occurring in that specific family to identify if those are a major role in why some relatives in that family develop MS and others don't. So we saw a family pedigree before, and here's another one here. We have 3 people in this family in the darker colours who have MS, and 3 people and the grandparents at the top of the family that don't have MS. So we look for families just like this to study. We initially start with families that have 2 or more relatives with MS, and we invite the relatives with MS and their parents to participate. Unaffected direct relatives of someone with MS that is over 50 years in age, And we asked these people to provide a DNA sample through saliva or blood to allow us to access their DNA. And then we use that DNA to study and identify the genetic changes occurring in the people with MS that are not in their unaffected family members. So this example family, we've not recruited the male in the bottom generation because he's under 50. So we can't be sure that he's not going to develop MS at some point in his life. But the two unaffected people in the middle generation are over 50, so we've recruited them, as well as the 3 people with MS in the family. And then we're looking for genetic changes shared between the people with MS that are not present in the people that don't have MS. So we may find 1 or more DNA changes in each family, but then our journey doesn't stop there. We need to work out how each of those changes affect the genes that they occur in, and then how this affects those cells inside the brain and the body for MS to develop, and how altogether this causes MS. So it comes back to this complex picture with the multiple players to identify how specific DNA changes is affecting each one of these cells in the development of MS. And we can do this by studying these changes directly in the cells made from people in these families. And that is what Ash is going to talk about next, which is our MS STEM study. So you can get involved with the MS family study, help spread the word. Maybe you know someone that has a family history of MS or multiple people in the family with MS. You can collect a flyer from our trade table outside, or you can come talk to me. There are a lot of contributors to this study, like many studies. In particular, I would like to thank the families that have made this study possible. We've got 5 families at the moment involved in our study, and each of them are leading to really exciting findings for us to pursue in the laboratory. Right, there's Ash.
Well, good afternoon. I will just continue from where Nick has left. So I basically take care of the MS Biobank, and this biobank is basically not a very similar to a blood biobank, which you have normally heard of. This is pretty much a research-based biobank, and the idea of the biobank is to use samples, human samples from people with MS, and try to study MS per se using human samples. So this is something, a very recent development which is developed in biomedical science in the last 7 to 10 years, and that is what we want to try and use and move away from animals and guinea pigs, which are normally done in basic lab research, and move more closer to humans and try to see what actually happens in humans, and then try to extrapolate or work from there to make better understanding of the disease and go forward from there and see what we can do. So the whole biobank basically is divided into there it goes. Okay, so the biobank has 2 parts. One part is recruiting participants, and now what we are recruiting here is slightly different than what Nick is doing for the family study. We are happy to recruit even individuals in the family who have MS, whereas Nick, as he said, that it has to be 2 or 3 for being a part of the family study. But we are not discriminating that part. We are just taking everybody in. We are also inviting people who are non-affected within the family, or anybody else who is also willing to come and donate, who has no family history of MS. We are welcoming them also to make a complete set of population of people living in a certain area. That's how we look for it. So this is the first part of the biobank. Now, in the biobank, as per today, we have more than 100 participants of MS people who belong to all the 3 types of MS: primary progressive, relapsing remitting, and secondary progressive. From these 100 participants, we have already created 50 stem cell lines on which Kelly and our group is actually working to understand and study how MS goes ahead and add some new insights into MS. I will talk about this part in the second phase, how we do that. Within the bank, we have mainly 3 types of cells. One cell, basically the first ones are which we directly collect from participants whom we recruit, that is the blood cells. And the second and the third one, which is induced pluripotent stem cells and the laboratory generated brain cells. Now this is a little different. These two are made in the laboratory. It has nothing to do with participants as such, but these are the main precursor for the second phase, which is studying MS. So that's the part which is more interesting for us, which is making iPSCs and making brain cells, which basically help us to study MS. So I'm sure everybody here in the audience have heard about what stem cells are here or there in news about regenerative medicine, stem cell therapies and everything, but what exactly are stem cells? So stem cells are basically very immature cells, and what gives them this specific ability is that they can divide forever. They are immortal in a way. And also they can differentiate or convert into different cell types. So that gives it a very, very good viability to work and study downstream cell types or different cells of the body if you want to do that. Now, the question which comes to everybody, which I've been hearing always, is are all stem cells same? No, all stem cells are not same. You would have heard of bone marrow stem cells or umbilical cord blood stem cells, which people nowadays preserve for the next generation and kids and for better life. But what we are looking is for induced pluripotent stem cells, or the last line, which is iPSCs. Now, these cells are basically cells which mimic an embryo, basically in a way that when the life was actually formed, when you or me were as an embryo, a single cell. So we are basically talking about science fiction movie. If you are, anybody is interested in science fiction movie and has seen movie, we are talking about not back to the future, but back to the past. So we are going back in time, taking an adult cell from a human being that is collecting your blood or anybody's blood and pushing them back to the stage when you were an embryo. And from there, we start working forward and trying to make brain cells, which is basically a replica of how your brain cells are behaving. So the more the number of participants we have, the more different kind of brain cells we have, which will basically depict me, X, Y, and Z person in the brain. So that gives us a very good population study-based analysis of how different brain cells work. Now, the advantage what we get is people having MS and people not having MS, especially in families, where we can specifically identify what is different in brain cells of people with MS, or what is different with in people who have not got MS, in this case of families. So that is a very strong potential. And this is just a schematic. What I wanted to show you what we do in the lab this is a picture of, this is a picture of the blood cells, what we collect from individuals, and this is under a microscope. If you put them under a microscope, our blood cells pretty much look like this. Now, on these blood cells, we give a cocktail of magic factors, and these magic factors basically transform them into cells. By 25 days of transformation, what we end up is a cell or a group of cells which basically mimic your embryo. So basically, you put them back in time from a mature cell back into a fetal state or an underdeveloped state. Now, what we plan to do in this whole thing is use these cells, that is the stem cells. So we start with the population, recruit families or individuals, make stem cells, and then with these stem cells, we can make various kind of brain cells, which we are looking at, maybe neurons, astrocyte, glial cells. And of course, it will help us in studying genetics from brain cells, as what Nick is talking about. As well as we can also try drugs, which we can never give to a human being to be tested, because we can't use humans as guinea pigs, but we can always use cells as guinea pigs and try whatever we want to do them. And it gives a direct correlation of how the drugs will behave when later on we move them into clinical trials and return some better therapy or better things to people with MS, and that probably would be something which we are looking forward in the next coming years. with that, I would like to thank my supervisor and with Kelly, with whom we are doing this work, Professor Bruce and other members of the community who have come forward and been very happy to participate and donate blood. If you are, anybody here is interested, please get in touch with me. There are flyers in your bag, and you can get in touch with me on a later time also. And of course, none of this would be possible with the fundings and every support from others. Thank you very much for your time.
So today we're going to talk about the Understanding MS Massive Open Online course, and I'm going to be talking about a little bit of what we found out so far in all of the research that we do around the course, which there's a lot going on, but I'm just going to talk about a little sliver of it. And then Jody's going to talk about how the things that we found in our research impact services and how we provide services and how you might think about doing that as a service provider, or even in some cases in a healthcare context. Let's see if I can get this to do what I want. Yay. So what is the Understanding MS MOOC, or Massive Open Online course? Well, lots of you in the room have taken the course, and even some of you in the room star in the course. We see a lot of familiar faces in the room. But for those of you who aren't familiar with it, it's a freely available online course that walks through various aspects of MS, starting at the underlying biology and pathology, all the way up to its impact on everyday life. And what's the goal of the MOOC? Why did we build it? What were we trying to achieve? Well, the main goal of the MOOC is to improve and increase people's understanding and awareness of MS, both in Australia and around the world. And so then the main goal of us studying the MOOC is to see whether or not we've actually achieved that. Have we met our goal? Have we approached that aim? And also, along the way, gain a better understanding about MS-related knowledge. So how do we go about doing that? How do we study the MOOC? Well, we do it in two major ways. The first way is that we run cohort studies around each enrollment of the MOOC, and that means that we ask a group of people who have enrolled in the MOOC to do assessments before they start the course, immediately after they finish the course, and then 6 months later after the course is over. And that lets us look at differences over time, so we can see whether or not people are actually learning anything in the course, fingers crossed. And the second way that we look at the course and we sort of study its effect is we gather information from people when they're actually taking the course. So just in the normal process of being a participant. And that's information that we gather around specific things that we ask people in the course, in discussion boards and other ways. And also just around engagement. So just seeing how people engage with the course. And so one of the main things that we've learned is that people living with MS know a lot about MS. I don't think that's a surprise for the people in this room. People living with MS are experts in MS. And so we assessed people's knowledge of MS before they start the course, and what we see is that people living with MS know significantly more than people not living with MS, but also, let's see if I can work the laser, I'm always terrible at the laser. What we see here is that people living with MS have higher than average knowledge of MS across the whole group, but also they're the second most knowledgeable group of any of the subgroups, second only to specialist healthcare providers. So they know a lot going into the course. And then when we look at how knowledge changes over time, what we find is that thankfully, the MOOC does teach people about MS. What we find is among people living with MS and those not living with MS, everybody learns more about MS and knows more at the end of the course than they knew at the beginning, which is great news after years of work. But among people with MS, we also see an uptick in some other outcomes. So we're also checking on a few other things. And what we see is that among people living with MS, health literacy, which is the ability to find and use health information and to navigate health systems, and self-efficacy, which is the belief in one's own ability to do things, also tick up after the course among people living with MS. And those upticks are less, are more modest than the upticks in knowledge, but they're consistent and they are retained 6 months after the course finishes. So does that mean that MS knowledge is related to those other outcomes that we saw that uptick into health literacy, into self-efficacy? Well, we're researchers, so we're the type of people that just can't let well enough alone, so we wanted to find out, and we guessed, we hypothesized that it would be. We've got MS knowledge here in this little flow chart, and it's connected by a bunch of lines to things like health literacy and self-efficacy and resilience and a few other things, because we thought they would probably be connected. And then changing MS knowledge would be related to changes in those other outcomes. So we took a look to see if we were right. And if we look at the data that came in from before the course started, so at baseline when people hadn't yet actually taken the course, what we see is that among people living with MS, their knowledge of MS is higher than people not living with MS, but their quality of life and resilience and health literacy is lower. So then we say, are those things related? And we took a look and we did some analysis, and it turns out they're not. So these things are separate going into the course. So then the next question is, does the change in MS knowledge that happens after you take the course because you've learned things, does that have an impact on the change in these other outcomes? Well, it turns out, no. There's no relationship between the change in MS knowledge and these other characteristics that we looked at, despite the fact that all of them improved. So that caused us to change our flowchart. So you can see here on the A side that we have the original hypothesized flowchart where there's a connection between MS knowledge and other outcomes. And then on the right, you can see a revised one where it's sort of MS knowledge is off by itself, and there are connections between these other outcomes, but MS knowledge is not connected. So despite the fact that MS knowledge isn't connected to these other outcomes, MS knowledge is important, and it's important because it's empowering. It allows you to make informed decisions, to ask informed questions, it allows you to pass on knowledge to others, and these are incredibly important things. So this is not to diminish knowledge in any way. I'm a humongous nerd, and I can't sing the praises of knowledge enough. But what it does tell us is that MS knowledge isn't necessarily the only thing you need. If what you want to do is change some of these other outcomes, some behaviors or skills, these are things that we might need something beyond knowledge to accomplish. And we think that the reason the course causes positive outcomes in things like health literacy and self-efficacy is because we've woven in other techniques. And that those techniques, in addition to providing information and increasing knowledge, probably would lead to those positive results that we're very proud of. So if we want to change behaviors, if we want to improve skills, information and knowledge isn't enough. We need other tools as well. And on that note, I'm going to pass it over to Jody to talk about what this means.
Oh, I've missed Kevin Bacon. I don't know, Francis Bacon. Kevin Bacon. It's a very funny 80s joke there. Hi. Anyone in the 80s should have had a little chat. So I guess when I found out this information, Susie was talking to me about the research, I thought, well, from a services perspective, we've sort of lived in this paradigm for a long time that we teach and teach and give knowledge, and that's what works. And so I guess being an MS nurse for 20 years, I had enough stories to know that that was probably not the truth. And that it didn't surprise me at all that people with MS had great knowledge coming into the course, and so like Susie, that wasn't surprising. So I guess it spoke to me about what we need to do in clinical services and in services as healthcare professionals and services as MSL. How do we sort of support people to build self-efficacy and health literacy and those other things? Now, I don't have the answers. I wish I did, and I'm working on it. But I think it sort of spoke to what is self-efficacy and how do people feel. To me, self-efficacy is when people talk about feeling in control. Feeling in control of their disease. They've got their plan, they have the right knowledge, but they also have the right team, and they also have the right resources around them. And how do we help people gather that sense of self-efficacy? Because if it's not just knowledge, where are the gaps? And if you think about self-efficacy, you think it's a product of your health story. We don't talk much about our health story, but it has such a huge impact on how much sense of control we have over our disease. I mean, your health story is, you know, how you grew up believing about taking medications is a classic example. You know, if you grew up with your mum saying to take Panadol, you'll be okay with medications potentially. But if you grew up with a person who's never taken medication, you're going to have anxiety around that. And so self-efficacy and our sense of control over things and power to be autonomous in decision making is a lot to do with what our values are in our health story as well. So helping people understand that can be really useful. So I had a look at the usual behavioural psychology, which is 4 ways to improve self-efficacy. I like the words, those 4 ways, 8 ways, 6 ways. But this one I thought really, this one was a sense of, you know, you feel a sense of power, you feel a sense of self-efficacy if you had a mastery of experience. You've taken it on, you walked that walk, run, roll, you did that 5 kilometres and you can do it. And so that's a great sense of feeling, I've done this once before and I know I can do it again. Vicarious experiences is another way that we have sense of self-efficacy as well too, and that's where the value of peer groups is so important, because if you can see another person have done it, you think, oh, they're like me, I can do that too. I mean, I listening to this morning, I thought I can even be an artist. So it's that sense of matching yourself with someone, and peer groups are really valuable for that. And to have those around you who kind of give you that sort of self-reflection. Verbal persuasion is another way that we have self-efficacy as well too, and that's encouragement. I guess I'm a very passionate MS nurse as well for a long time, and I think nurses are very good encouragers to people to be able to sort of go for their gold and go for their goals, but also other people around you, and that's where setting up right from diagnosis, those people around you are going to be, you know, right in there with your values and what encourages you. And again, I'll use the art story, that person who is cheering you on right from the start about doing that art, doing what's important to you, to give you a sense of control and self-efficacy in your life. And then, I guess, being psychologically or psychologically prepared. And we all know that, you know, when you're depressed and when you're fatigued and when you're feeling all that sense of overwhelm, that readiness to feel empowered and a sense of control, I'd probably put that at number 1, because it's just so hard to kind of reach that level when you're right down there, and sometimes that's just where we need to start. And I think when we think about providing services, even when we think about doing research for people with MS, we should be looking at how does that reflect in this area as well too, because that's, you know, the people who have the best outcomes in MS are certainly the people who have a strong sense of self-efficacy. Then in terms of health literacy, Susie defined health literacy, but there's very good evidence to suggest that those people with a low health literacy do worse in health. And that's usually defined, we often think of health literacy as the way that something's explained, that it makes sense to people. I like to take it a step further and think health literacy is also how people make sense of it to them in their personal context. And that's often a one-to-one conversation. It's often that one-to-one sort of sitting down with someone to sort of really, how does that actually fit in with me? How does that fit in with the symptoms I'm experiencing? How does that fit in with how my values and where I live my life? And so I think that's a real aspect of health literacy that's often ignored. You can have nice, pretty pictures and explain things really well, and just listening to the beautiful explanation of genomics there was just, you know, that was just magnificent from health literacy, thank you. But I think we've got so much better at that as healthcare professionals, but there's a long way to go to that next level that people are able to sort of embed it in what that means for them personally. And the other thing I think a lot about when I think about health literacy is that aspect of the health literate organisation. And that's about, you know, that sense of self-efficacy comes from knowledge, but also knowing what to do when. And that's a little bit about not what you know, but who you know. And I'm sure my children will say to me that's going to be on your avatar, Mum, because I always say, it's not what you know, it's who you know. And so it's about sort of helping people understand how to navigate the systems, because that's so important. And I think Heathrow Airport is a classic example of how they actually improved the literacy of that airport, because you would get there and be completely confused, and then some brilliant person came along and made one whole section that's all purple and one's all orange and one's all blue, and just made it so much easier to navigate. And healthcare systems are really complicated. They're not literate systems. And that whole concept of what to do when and how to do it. So that's another part of health literacy that I think is really important to be able to incorporate with knowledge, and they fit together so nicely, and they can really be communicated with each other as well too. You need to have that knowledge. It's so important. So it's such a part of our jobs as services to do that, but it's also able to give you the rest of that health literacy, so you feel like you have the language that you need and the resources that people with MS need. So that's really the call to action for us as services, to be able to support people doing that, taken from this wonderful research where we could work together and learn together, which is the best way to do it.
Absolutely, and if you are interested in getting involved at all, you can enroll in the Understanding MS Mooc and take part in research yourself. You can spread the word about the course, or you can help us create new short MOOCs, which we're working on 4 separate short MOOCs, and if you're interested in taking part in any of that, please come talk to us at the trade tables. Thank you so much.
So, Andrew and I both work for MS Australia, and I hope by now you've met our board president, Des, and I hope by now you've met our CEO, Rowan Greenland, and our new head of advocacy, which also scoops in comms, Jeremy Henderson, sitting over here if you haven't. Often when we present, I show this diagram because I know a lot of people get a bit confused about MS Limited and MS Australia, MS Western Australia, etc. At a glimpse, I hope this will show you our state, our national, and then our international connections. So MS Australia is the member organisation of the Multiple Sclerosis International Federation, and with our research colleagues, the International Progressive MS Alliance. I hope at a glance that's shown you how the MS family all fit together in Australia. A couple of examples of the basic information that we provide to decision makers about MS. We've got lots of evidence-based material available to us. We can tailor responses to just about any inquiry we get at MS Australia and to suit just about every lobbying and campaigning opportunity. Much of this evidence comes from the researchers in this room, so another plea to support their work. Without your involvement, the researchers' work that gets synthesised, that helps our advocacy and lobbying. Here's a great example. You'll all be familiar with the Menzies Health Economic Impact of Multiple Sclerosis report. We then turn that into a key facts and figures document. That's now been translated into 10 languages. It's available on our website, and as I said, it's come from the Health Economic Impact report, which we're hoping that the Menzies will run again soon. How do we know what to advocate for? How do we identify the service gaps? When we say service gaps, we're talking about federal government, basically, service gaps, NDIS, aged care, healthcare, and so on. How do we know what your needs are? So this just is showing you the top 11 priorities, which were results from a major priority survey we conducted last year, mentioned earlier today by Viv. There was about 3,000 respondents, plus Andrew and I conducted some focus groups, plus we get ongoing feedback from people with MS, probably from some of you in the room, plus from our state colleagues, people like Jodie and her teams. These results informed the development of our strategic plan. They also informed, for example, our 2022 election commitments document. This feedback also steers our responses to government and agency inquiries, and it tells us what to focus on. Here's our strategic plan at a glance. It's easy to find on our website. We're not going to read it out to you, but just as you can see the main headings across the middle of the page. So all of that material gets synthesised into a document like this. Our advocacy team are primarily responsible for the first bit, people living with MS. their carers, friends and family. We're also responsible for the education and awareness bit and the advocacy bit. And because we can't help ourselves, we involve ourselves in all the other bits as well. I'll hand over to Andrew. I will.
Hello, here's the 2022 election commitments document. Just so you can see the link between the 7 issues we asked politicians to commit to and what you, the MS community, told us. Thank you, Debs. And what you, the MS community, told us you need or you would like from a political perspective. If you can remember the photo of people in the bottom of that brochure, I'll come back to that in a moment. Next one, please. Alliances are critical to progressing our advocacy work, as you would imagine, and we can't do it all on our own. Many issues affecting people with MS affect many other folk in our community. So we've got a strong link with other alliances of like-minded organisations that are represented. Next one. These slides set out our major advocacy tools of trade, and as Senator Wendy Askew was here this morning, she co-chairs, as we know, our bipartisan parliamentary Friends of MS, along with Senator Deb O'Neill from New South Wales. Both of these folk, as Wendy Askew shared this morning, were re-elected. Here's a ripper. A recent example, survey of people with MS told us that we all wanted better access to MS nurses. Surprise, surprise. MSA commissioned a study from the Menzies, and the report from this study was launched at our recent research conference in Hobart just in April. The working party of the report partners is now planning advocacy activities to address each of these recommendations. There's an executive summary document in your billy bag that you have with you, so read it with much zest and enthusiasm, please. Next one. This is just sharing for you recent and current work and some examples of them. Given the nature of time and pressure I'm under, Des, I won't read them out for you. This document is obviously available. It'll be, if it's not in your bag, it'll be on the website. I'll send it to you as participants here today. How to get involved? The next one. You've done that for me, thank you. We are the B1 and the B2, or the A1 and the A2 team, as you guys who know us know. To finish, how you can help us? I can't read it that far, it's too far away, but I can see it here. How to get involved? You can keep an eye on the website, follow us on Facebook, Twitter. Always happy to get feedback, so if you want to give us any feedback, email him. He's the Giles. And of all things, most importantly, given this Monday coming, for me it's 2 things. One, it's World MS Day. It's also my youngest daughter's birthday, so I've got 2 hats on Monday. That's why I'm going to fly out in a minute. But do what you can to support World MS Day on Monday. And it's very nice to see people on the happy end of the island in Launceston, given I have a Persuasion being a coaster. So thanks very much, everybody.